Okay, this section of the course addresses how actually impulses travel along nerves. So we're dealing with 4.1.2, C, D, and E. And in this we talk about, there's going to be quite a lot of new vocabulary, so keep your pens handy. So resting potential, action potentials, we'll come back across myelin again, and then these things called voltage-gated sodium and potassium ion channels. Now, just to give you some context of what this section is about, what we're dealing with here is one chunk, one section of a neuron. Here's a sensory neuron diagram that we used before. So we're dealing with one cross-section, one part of this. And if you can imagine the work you're about to go through and what you're going to try and understand occurs in multiple stages. So at every place along this neuron. So this is about the transmission of the impulse from the receptors under the skin all the way through to the synaptic endings. And we're going to look at what happens just in one slice of that axon. So to start with, this is the animation that I'm going to use to try to explain what's going on. And just to familiarise you with what's on here, this is one section of the nerve, and we have on the outside in purple is the outside of the nerve cell of the neuron or the axon. The blue area is at the inside, and this area in the middle then is the plasma membrane with the pumps in the channels that are needed to try to transmit this impulse, this electrical signal along the nerve. Imagine in this animation that the impulse is being transmitted as per this diagram above from the right side to the left side. Doesn't really matter too much, but you may see the relevance later on. Okay, first of all, just so you know what's what in this animation, the green blobs that you see above are going to represent sodium ions, and hopefully you remember from your GCSE chemistry, sodium ions are positively charged, and the purple blobs that you see in the animation above represent potassium ions. So these are charged particles. They're both positively charged. Now this is important because the way that a nerve actually works to send the impulse along is reliant on differences in potential, and to gain some kind of potential or a potential difference, and again you should remember this from your GCSE physics this time, potential difference, otherwise known as a voltage, you must have charged particles, you must have ions. So a potential difference, as the name suggests, is a difference in potential, a difference in the number of charged particles you've got on one side of a boundary, in this case a nerve membrane, compared to the other side. So what we have here at the beginning, before the nerve impulse arrives at this section of the axon, so remember we're just dealing with one area here, is that this nerve is at rest. And as you can see, in the resting state we have lots and lots and lots of sodium ions on the outside of the cell. We have many more potassium ions on the inside of the cell. There are some potassiums on the outside and some sodiums on the inside, but generally speaking there are more sodiums on the outside than there are on the inside, and more potassiums on the inside than there are on the outside. You may also notice that there are more sodiums here than there are potassiums here. So we have more positive ions on the outside than we do on the inside. Because we've got more positive ions here than we have here, this gives us a difference in the potential, a potential difference, a voltage across this membrane. You need to be aware of the value of this. I'll just write it down on the left-hand side. And so to begin with, at rest, this nerve has a potential difference of minus 70 millivolts. If you like, the outside is more positive than the inside, or the inside is more negative than the outside relative to each other. This number will change as we go through the different stages of this process. Now, in order to achieve this state where we have a more positive area on the outside, we use this guy on the right hand side, the sodium-potassium pump. I'm just going to start the animation, and you'll see what happens with this pump working. That it pumps out 3 sodiums for every 2 potassiums that move in. Just going to wind that back so you can see it again. So 3 sodiums move out and 2 potassiums move in. So that's our sodium-potassium pump. And you can see the grooves on the inside there that represent the movement of those ions in and out of the nerve's axon. So if that repeats over and over again, then you can imagine that we end up with more sodium on the outside and more potassium on the inside because of that 3:2 ratio. And that's how we achieve our resting potential. So that's another key thing for you to remember, the sodium-potassium pump. Okay, when this section of an axon is ready to do its job, its part in the transmission of an impulse, this is what happens. It receives a signal. Remember, we're traveling from right to left. Slightly counterintuitive because normally you would read from left to right, but just imagine we're going from right to left to suit the diagram that we've got above. So this section of the axon is going to receive an impulse from the section of the axon that's next door to it on the right hand side. Imagine there were the same diagram again, but over here. And there are some changes in the potential over here, and that causes the following events to occur. So first of all, when it detects that change in potential, the sodium channel opens and in rushes the sodium ions. This sodium channel needs a proper name, and we're going to call it a voltage gated sodium channel, and that's because it works when it detects changes in the voltage of the membrane around it. So I'm just going to wind that back again and play. So the sodium channel opens, that's stage 1, and the sodium ions rush in. Now they move in from an area of high concentration to an area of low concentration, and the biology part of your brain should be telling you, I've recognized that as a concentration gradient, but we need a bit more detail, and we call it an electrochemical gradient. And we call it an electrochemical gradient because there is a movement down the chemical gradient from high to low concentration, but also there's also an electrical gradient between the two areas. So an electrical gradient and a chemical gradient, we put the names together to make an electrochemical gradient. So the sodium has rushed in. All those positive charges, those ions, have now moved into the inside of the cell, and this affects the potential difference, the balance across the membrane. So we've now moved from minus 70 to plus 40 millivolts. So now we have a very positive area inside the neuron and relatively much more negative outside. This is what's just happened is something called depolarization. So the depolarization was the movement of those sodium ions inside the nerve cell through the voltage-gated sodium channel. So our neuron has now become depolarized, the sodiums have moved in, and in response to that, the knock-on effect is that the potassium channel is then going to open as well. And as you would expect, the potassium ions that we've got inside the neuron move down their own electrochemical gradient, and they move from inside the neuron through the voltage-gated potassium channel to outside the neuron. So the sodiums have moved in, and then the potassiums move out, and this part is called repolarization. So depolarization was when the sodium ions moved in. Repolarization is when the potassium ions rush out down their electrochemical gradient. So many potassium ions rush out to the outside that we actually then achieve a potential of minus 80 millivolts, a little bit higher than what we started with. And we call this an overshoot, and we'll come back to that a little bit later on. Essentially, this part of the neuron has now done its job, and what we've just seen is what we call an action potential. So the action potential has passed through this part of the neuron, and you can imagine again if there were the same diagram again, but to the left hand side over here, that the same series of events would then happen on the next part of the neuron, and again and again and again and again, and there's a kind of domino effect along the neuron until it reaches the synaptic ending. This part of the neuron needs to recover and return back to its resting state, its resting potential of minus 70 millivolts, in order to then receive another action potential if it needs to. So we need to get back to our original state, which, if you remember, was sodiums on the outside and potassiums on the inside. You may be able to see what's going to happen next. So the potassium channel closes, the sodium potassium pump opens up, and it transfers 3 sodiums for every 2 potassiums, and there's this continual movement of those ions until the potential difference across the membrane returns back to resting potential, which is minus 70 millivolts. Important finally, just for me to re-stress again, what you've just seen is what happens in one section of this neuron, and the impact on that is the next section of the neuron, so say here, then detects the changes that happened here, and the same series of events happens again, and then here, and then here, and then here, and it moves all the way along to the end until it reaches the synaptic endings.